Good evening, my dear new students. Can you see PPT? Have you good sound? Tell me, please.
Yes, doctor.
Okay, thank you. Okay, I introduce myself. My name is Harayeva Zaira, and I'm Chief of Department of Microbiology, Virology, and Immunology. And you will study at our department some subjects like immunology. We begin with you, and you will study immunology all this semester, and we'll have that shot at the end of semester. At the next course, at the third course, you will study at our department microbiology, virology, and you will have exam and pharmacology. Pharmacology is subject of our department too. And at the sixth course, when you will be very near graduated students, you can choose immunoprophylaxis. In this course, you can study some modern information about vaccination, prevention, serum, base effective prevention, and prevention with immunotropic different drugs. All this material is very interesting and very important in understanding of clinical some aspects. But we begin from immunology. First of all, what rules we have in our lectures. I have one lecture on 2 weeks, and of course, I can't give you all the information that you need. But I ask you to have books. You can take electronic version of our book on immunology from your teacher. You can take manual from your teacher in electronic version too. Plus, all this material you need to have. And please read more sources in the internet, maybe some books, and try to compare, try to analyze, because immunology is basic discipline. It's very important in understanding of your future subjects like pathophysiology, pathanatomy, and you will construct your good base for future clinical disciplines. I ask you to join to this conference only under your name. Today, I can see that among our students, I can find iPhone or Samsung, but unfortunately, your teacher can't understand who is it iPhone, and I will not join again somebody without a name. It will be very good if you add to your name number of your subgroup. It will be much more easy to teacher to find your name and to put that you attend this lecture. How I can check your attendance? Don't write your names into the chat. I don't read into the chat. Only can if you have some question, okay, you can write it in the chat. But I can see this chat only before our break and before the end of lecture. During lecture, I can't every minute see what maybe you write to me. And if you have some question about lecture, about material, please collect all your question and ask me at the end, before the end. What I ask you to know that I take photo twice before break and before ending of our lecture, and give this photo with all lists of students to your teacher. I'm only your lector. Your teacher will be Dr. Nothing or Dr. Narina, and all these pictures they will have. Thus, if you write not your name, but for example, um, your phone, um, your teacher can't understand who is it, and you will be without scores of your for your attendance. I hope is it clear? Is it clear for everybody?
Doctor, like we have only capacity of 100 students. So other students who are unable to join, what about their attendance?
I have right now 100 participants. Thus, maybe Zoom can't join all other students to my conference. But if the students can join at the next hour lecture, it will be next Tuesday at the same time. They can join. I only can do this style. But I have some students under not their name, and maybe if some of them try to join under a new name, it gives for us so not a good situation with joining. Let's begin and try to understand what we will do next time. Our first topic is about innate immune system. Innate immune system is a very interesting first defense of our microorganism. And we begin as usual from definition. How we can name this interesting system, defense system? Innate immune system is the same as congenital immunity because this is genetically encoded system of real first defense. Natural immunity, non-specific resistance is all the same names of one system, like innate immune system. We can't, for example, delete innate and use in our explanation immune system, or we can delete congenital and only tell immunity. And I can't understand you in this if you try to give me so explanation, because innate immune system and real immune system, it's the two different system of defense. Thus, adaptive immune system is the same as immune system or is quiet immune system. Don't miss these names and use it in right way. Introducing, first of all, we must understand that many years, many immunologists, they thought that maybe innate immune system it's only about phagocytosis at all. Phagocytosis it's something when some process when macrophages can kill some bacteria, fungi, or some another on cells. But now we understood that innate immune system is very interesting, very strong and very important system. And new roles that innate system acts it's like regulator of adaptive immune system. Because it may be like activator, and you will see some examples and you will understand in what examples for example cell of innate immunity will be at the beginning of immune action, immune response, and sometimes controller, because sometimes in our organism we need to depress activity. Thus, please put your attention here and try to understand that the innate immune system has different regulatory mechanisms of influence. Uh, First of all, what we must understand, this is genetically encoded natural disease, natural resistance and features of innate. Azad, if you don't listen to me, please go and rest. And maybe some another students who would like to join. Sorry, ma'am, it's my mistake. Sorry, ma'am. Okay. Features of innate immune system are very interesting and very important in understanding of function of this system. First of all, non-specific factors are involved in protecting the body against any antigen regardless of their nature. What does it mean, all these sentences? What does it mean, non-specific factors? We must understand that our cells of innate immune system recognize their targets, but what they can recognize, they don't recognize antigen. They recognize some markers on the cells that gives information for our innate system that this is wrong cell. But it's not antigen, it's like markers of some group, like oncomarkers, or for example, some pattern for all gram-positive bacteria, or some marker of all gram-negative pathogenic bacteria. Thus, innate immune system can defend us without recognition in detail, without recognition of antigen, but with recognition of some groups of molecules, which we will name like pattern of some damage, pattern of some infection, pattern of something wrong processes like oncology. What's the next very important feature is that the innate immune system has no immune memory. In contrast, immunity, we know about vaccination. We know that if we, for example, in our childhood caught some infection, we can for example, scarlatina or some ruby virus infection. We all our life have resistant to this infection, but this is adaptive immunity, not innate. Innate immune system has no immune memory at all. The third, the innate immune system has constantly active mechanisms and some mechanisms that can be activated after contact with something wrong, like pattern. What's the differences with adaptive immunity? Adaptive immunity can't be constantly active. Adaptive immunity will be activated only after contact with antigen, not constantly. Thus, please write all these features in your papers and try to compare innate immunity, adaptive immunity. Innate immunity recognizes patterns. Adaptive immunity recognizes antigens. Innate immunity has no immune memory. Adaptive immune has some mechanism of immune memory. Innate immunity has constantly and activated mechanism of action. Adaptive immunity has only activated after contact with antigen mechanism of action. What's the classification of innate immune system? You can see this classification according to their nature. And if we try to analyze, we can find some examples of constantly active component and components that must be activated. First of all, anatomical physiological barriers, very easy understandable form of part of innate immunity that we all our life have a skin, thanks God, or some mucous membrane that very good defend us from many microbes, for example. Antimicrobial proteins in tissue liquids and into the blood, cells of innate immunity. We will study with you and normal microflora of organism. I put your attention to here, to this microflora. We only at this course of immunology we can understand that normal microflora is part of innate immunity, but we will study about this in details in course of micro. Now we know this classification and study these three first points. First of all, about anatomical physiological barriers. Anatomical physiological barriers may be like our skin, mucous membrane, some mucous membrane of gastrointestinal tract or a genital tract in oral cavity, some mechanism in respiratory tract like mucus, like ciliary action. We must remind about, for example, blinking teeth and some lysozyme in all these liquids. I put at the slide for you some explanation, but it's very easy material, and if you try to remind all your knowledge from biology, from anatomy, from physiology, you can do all your answers more good. For example, we must understand that skin is not only mechanical barrier, that in the sebaceous gland we have some different active proteins, and they give for us additional defense. We must understand that it will be this physical chemical barriers will connect with gastric juice, with production of some acids like acetic, formic, lactic, and all these physical chemical barriers are very important, and we can understand their influence when somebody has, for example, pathology like low acid gastritis, low concentration of hydrochloric acid in gastric juice. All these patients unfortunately suffers from many infections. Why? Because they have low pH in this gastric juice, and they can take very easy many infection with food, with water, and this is like very good example. Or, for example, when we have some wound and this damage of skin may be complicated by infections too. About antimicrobes protein, lysozyme may be the most interesting, and maybe you have studied about lysozyme, and you have some information before, but we must know that old name of this enzyme is muramidase, and this is enzyme that can hydrolyze cell wall of bacterial cell. Every bacteria has some structural elements, but some of the structural elements are necessary for living, and for living they must have cell wall. Lysozyme, it's like scissors, like enzyme scissors that cut the cell wall to parts, to small parts, and after action of lysozyme, bacteria will be killed. Lysozyme is synthesized producing by macrophages, neutrophils, and other cells. And in a very high concentration, in the most concentration, it contains tears, just a little less in saliva, just a little mucous membranes of the respiratory tract, in blood, in the lymph, in the milk, and in the gastrointestinal tract, in all our mucous membranes, of course. Before antibiotics, we must know it. In pharmacy, people can buy lysozyme in bottles, and in that times, doctors use this lysozyme like antimicrobial drugs for example, treatment of some wounds or some infection of throat and others. Another antimicrobial proteins which we must know, beta-lysins. Beta-lysins, very interesting active proteins that are produced most of all by platelets. They are in high concentration. Please switch off your sound. Beta-lysins elevate permeability of cytoplasmic membrane. This is CPM, cytoplasmic membrane, and cells will be killed too. But it's not so strong with so strong effects like lysozyme, just a little less. In every liquid, in blood, in tissue liquids, we have beta-lysins. Acute phase proteins like C-reactive proteins like mannose binding lectin, they take part in covering of bacteria and preparing bacteria to phagocytosis. This process is named like opsonization. Opsonization is covering of bacteria by our different proteins, and this covering will elevate efficacy of phagocytosis. Macrophage can recognize the stages much more easier than without this protein. All these acute phase proteins, they are produced in the liver, usually in response to some damage, to some infection. Thus, we can use this concentration of C-reactive protein, mannose binding lectin, as markers of inflammation, and we do it when we try to analyze the results, blood results of patient. Usually, we try to find, for example, CRP, C-reactive protein. Mannose binding lectin takes part into the activation of complement system, and we will see in one of pathway of activation role of this protein. Another proteins, properdin. Protein is that protein that may be in serum in our blood, uh, involved in some immunological reaction, but activates a complement system too. It's one of this group of proteins, BDP, which we can find in alternative pathway of activation of protein. Fibronectin is universal protein of serum, and we have fibronectin in some concentration in all our fluids. It takes part in opsonization of antigen 2, and opsonization, as I told you, do phagocytosis more effective, and I hope you catch this sense. Thus, among all this classification, we begin from anatomical physiological barriers. We put our attention to antimicrobial proteins, and into this group, it was lysozyme, it was beta lysins, it was C-reactive protein monobiotindectin, it was properdin, fibronectin, and in this part there is complement system. I know that in the course of physiology you have some topic with complement system, but I ask you to add some information to that you studied, that material, because in immunology we must study 3 different pathways of activation, and we must understand results of activation with accent of antimicrobial defense. But please try to understand this part, complement system. What is it, complement system? Complement system is interesting system that consists from more than 20 different proteins. All these proteins work together, and as a result, they construct a membrane attack complex, MAC, membrane attack complex, and cells which were targeted for membrane attack complex will be killed. But is it the main result, the most important result, killing one cell? Of course not, because in all these active components of complement, who draw at my slides? Maybe you are in kids garden right now, or you are students of university. Okay, I try to explain you, and you don't give me this opportunity. Some fragments of complement during activation can act like chemoattractants. Every chemoattractant is very important, like biochemical signal. Biochemical signal that give our organism, our cells, and other cells, all other organism, biochemical signal SOS. In this local, we have some problems. This high concentration of chemoattractant, this gives for a sense. Thus, complement system is some interesting regulatory, first of all, regulatory system of defense. And in addition, some of these chemoattractants may be like opsonization markers. I told you twice, and now I can tell you the third time, that opsonization is covering degradation of surfaces of invaders by this protein, and after this, covering these markers, these targets will be phagocyted much more easy and much more faster. Thus, complement system has two free function. The third, killing of some target. The second, inducing of inflammation, and the third, abstinization. As a result, activation of phagocytosis. Pathway of activation of complement system. We must know three different pathway of activation. At physiology, you have studied, as I know, only three or only two: alternative and classical. Don't draw at my slides, please. I will write all the names who try to drag my slides and give this information to direction that you don't give me opportunity to read your lecture. Are you kids? Alternative pathway, classical pathway. To these two pathway, we add lectin activation. We begin from alternative pathway of activation. First of all, put your attention to activation complex. If you understand components from activation complex, you will understand what factors may be the most important, what sometimes results will be more important for all organisms. You can see here LPS. What is it? Please write it. Lipopolysaccharides. What is it? Lipopolysaccharides. Lipopolysaccharides are combined interesting carbohydrates that are components, most of all, of gram-negative bacteria. Thus, LPS, this is like markers of gram-negative bacteria. Thus, our first conclusion from this activation complex is that alternative pathway of activation, this is activation most of all against gram-negative bacteria like Escherichia coli, like Salmonella, like Shigella, like Klebsiella, and many others gram-negative bacteria that, for example, for example, they can cause dysentery, salmonellosis, klebsiellosis, typhoid fever, and many others. But we put our attention to the second component. Second component, BDP. BDP, this is some protein about which I put your attention here. This properdine, BDP proteins, they are in small concentration in our blood, in our liquid, tissues liquid, and here they connect with cells of gram-negative bacteria in presence of magnesium. Thus, we must have good concentration of magnesium in our blood, in our organism. As a result of organization of this complex will be activated C3. C3 is classical protein of complement system. After activation, this C3 will be like destroyed into two parts. C3A will be like chemaattractant, and C3B will be a real active enzyme. sound, please. If you can't listen to me, go and rest. C3b, this is active enzyme that find its target and try to activate it. Thus, please understand again, alternative pathway of activation begins from formation of this complex. LPS, this is part of cell wall of gram-negative bacteria. In the presence of high concentration of magnesium and BDP proteins will be activated C3 components that after activation will be C3A part and C3B. C3A gamma-attractant, C3B enzyme that will activate C3-5, C3-5, C6, 7, 8, 9 components. And as a result will be this membranated complex and killing of some cell. But what cell? This cell. Okay, let's continue. Have you good sound and can you see my PPT here?
Yes, ma'am.
Mm-hmm. I ask you again, don't try to name into the chat. I do end of lecture and give this information for your teachers. Thus, much better will be if you try to join under your names and add your subgroup. It will be better for teacher because they will be for them it will be easy to find your name and to put some scores for your attendance. Okay, let's continue. We interrupt with you when I explain you about alternative pathway activation. I ask you to analyze activation complex, and if you listen to me, you must understand that alternative path of activation of complement system is part of antibacterial mechanism of defense. Most of all against gram-negative bacteria, because this LPS lipopolysaccharide is marker like pattern of gram-negative, most of all gram-negative bacteria. But killing of this gram-negative bacteria is not the main result of activation. We must understand that during all this activation chain will be inflammation and axonization, thus elevation of phagocytosis. Classical pathway activated by complex antigen and antibody, and maybe it's very good because antigen may be real nature. It may be oncological cell, it may be gram-negative bacteria, it may be gram-positive bacteria, it may be, for example, some damaged cell, our cell that may belong to us, but find in this complex something that limit us, because antibody must be in our organism. But if this antigen comes first time in our body, we have no any antibodies, this classical pathway will be not realized. Thus, classical pathway of activation will be only for antigen that comes rapidly, second time, third time, not first time. The results will be the same because as the results of formation of membranated complex, this antigen will be killed, and in addition, inflammation will be, axonization will be. this classical pathway, antigen antibody. It begins from component, C1 component, and this is difficult organization of some convertees. You don't need to to understand it in detail. You must understand in general that after all this activation will be killing of this wrong cell and inflammation and abstinization. This two pathway of activation you have studied in physiology, but we begin with you from what? That all this physiology knowledges you mastermind and add something important from this course. And we add with you lectin pathway. What is it, lectin? Lectin is complex carbohydrates. Carbohydrates are like some markers of most of all fungi because carbohydrates can be on our cells. It's patterns of some microbes, but among all microbes, most of all in the big concentration on fungi. And this mannose is a pattern of lectin, uh, pattern of ron, some not our cell. To this mannose and to cell that has this mannose connects mannose binding lectin. About this protein, we will start to review when we this slide try to understand mannose binding lectin in the group of acute phase proteins. And as a result will be activation from this fourth fragment after second and after the same chain. As results are the same: killing of some wrong cell, abstinization, and inflammation. Thus, please remind our classification: anatomical physiological barriers, antimicrobial proteins. We have started to review these two first parts of innate immune system. And we have cell of immune system. Cells of immune system, the last part of our lecture. What cells of immune system we must know, and what will be general information for all cells, how they can act, what's their activation. This is very important points which are real main part of our lecture. Cells of innate immune system. First of all, this is a professional phagocytes group: macrophages, neutrophil, natural killer cells, NK cell, and NKT cell. And in addition, it may be antibody-dependent killers. About each this type of cell, in brief words, I need to understand you. But first of all, we must understand how these cells recognize their stages, and they recognize patterns. Patterns are not the same as antigen. All patterns are alien, conservative structures. They don't change from one mutation, and pattern may be divided to two groups: PAMS and DAMS. With PAMS is pathogen-associated molecular patterns, and DAMS, damage or danger-associated molecular patterns. One pattern will be, for example, in each gram positive bacteria. One part will be for double RNA connected viruses, or for example, one pattern will be for all fungi. Cells of innate immune system recognize group, not antigen, but all group for this recognition of these conservative structures. After pattern binding results, what will be? Increasing of activity of cell of innate immune system in the form of increasing of phagocytosis and efficacy of phagocytosis, secretion of some antibacterial peptides in, for example, lysozyme, beta lysins, complement, some complement protein, processes and presentation of antigen, but this is beginning of immune response and development of immune response. Whereas innate immune system is in the beginning of immune reaction, and we must understand it. We begin from macrophages. Macrophages are very interesting, powerful, very strong cell. Three stages of macrophage: resting cell, non-activated, prime cell. When some chemoattractants knocks to the cell, okay, you must be activated right now because we have some SOS signal from that locality. This is prime cell and activated cell. Activated macrophage is very active, very powerful killing machine. Like here you can see, because it produces reactive oxygen species with its free radicals, many aggressive enzymes, and all these enzymes and all these radicals, free radicals, are very aggressive for example, target, and all this help to phagocyte target. Stages of phagocytosis I don't repeat with you because this is you must know from biology course, but we must understand that others macrophages function we must know, because this is antigen presenting cell, garbage collector tool, and very important and very powerful producing of some cytokine cells. Cytokines is like mediators, regulators of immune system, and we will study with you at the second lecture some information about cytokines. Thus, macrophage is maybe in different stages. Macrophage can phagocyte target. Macrophage is antigen presenting cell, garbage collector, and regulatory cell. Neutrophil has some similar properties with macrophage, but we must understand that this cell have short living time. Short time, and after apoptosis it will be killed. Neutrophil exit into the blood, and into the blood they can be activated. In activation form, it's very similar with macrophages. Natural killer cell and NK cell, they have another killing mechanism, granzyme perforin. Perforin is like a complex that will be in a target cell, and like a hole into the cell. Through this hole, granzyme enters into the target and leads to apoptosis, to death. NKT cell has very similar cell with NK, but has interesting regulatory aspects of this function. Sometimes destroying or function of NKT cell leads to autoimmune processes, to sensibilization, allergic reaction, for example, bronchial air asthma, and we must know that the NKT cell sometimes is very interesting markers in understanding of pathogenesis. And our last slide about K cell, antibody dependent killers. This cell has only one type of receptor that connects with excitosis with killing of targets. It only Fc receptor. Fc receptor is a receptor for antibody. Thus, the cell antibody only through antibody can connect with targets, and only through antibody will be like linking with target, and cell will kill these targets with granzyme perforin pathway 2. I put for you some additional literature which we can find on internet freely without any payment. Please try to read more about immunology. If you have some question, ask me right now. Don't write your names into the chat. I don't read your names. I repeat it again and again. Right now, I take photo. And if you have some question, I will be ready to answer you.
Ma'am, will you send this PPT?
Yes, I will send this PPT to your teachers, and you can find this PPT for your teachers. Of course, all my PPT will be yours PPT. Okay, have you another question? No?
No, ma'am.
Okay.
No, ma'am.
Okay, good luck for you, and thank you for your attention, and I hope we will have some good results in our starting of immunology. We'll begin with you. Bye-bye, and be healthy and happy. Bye-bye.
Thank you, ma'am.
Bye.
Thank you, ma'am.
Thank you, ma'am.
Thank
Thank
you,
you for the
ma'am.
nice class.
Dasvidaniya.